little bit on the rationale for using genome editing to achieve that goal. So first, in terms of the natural pathology of the disease, HIV itself targets the CD4 Tcells. These are a particular class of immune cell, and the loss of these cells leads to full-blown AIDS. So the CD4 Tcells are a key cell in the immune response necessary to control infection, not just of HIV, but in fact of any infection. And CD4 counts correlate very closely with disease progression. So falling CD4 counts ultimately are an indicator of the immune system's loss of, if you will, of progress against the virus, and ultimately they will fall to a level that will allow full-blown AIDS. So the idea is that we use genome editing to make a subset of CD4 T cells resistant to HIV. And so we don't plan to make the entire CD4 population in the patient resistant, but we plan to make a subset of these cells that are resistant. And the concept is that the protected CD4 cells, so those that actually no longer have the CCR5 co-receptor, and therefore HIV can't enter those cells obviously can't support new HIV replication. So the number of targets that HIV has to infect and produce more of itself is reduced. The more resistant CD4 T cells that we have, therefore, the lower the HIV levels would be. So obviously if HIV lacks targets to infect, then it can't infect those cells and can't give rise to new HIV levels that can go infect new cells. The protected CD4 T cells can also support or even reconstitute the immune system. So because HIV is no longer removing these cells are permanent or persistent and can allow the immune system to recover to support immunological responses to infection. And in that process, we have the potential to support an anti-HIV immune response. So in other words, that these protected CD4 T cells can not only in general help the immune system, but more specifically could support the immune system's ability to recognize and ultimately eliminate cells that are infected with HIV. And the proof of concept as to how this might play out is provided by Timothy Brown and his situation as an HIV-positive individual who developed an AML, a cancer, that was treated with a bone marrow transplant with cells from a donor who had a natural mutation in CCR5, so-called CCR5 delta 32 mutation, in both copies of the CCR5 gene. And so the cells that he received had no CCR5 on their cell surface, and he's remained aviremic now without the requirement for being on antiretroviral medication for over 5 years. And so this suggests a path for eliminating CCR5 as a target in appropriate cells to achieve a control of HIV.